Okay, so we're looking at development, basically how you start from a sperm swimming into an egg. Like, how do we start out as one cell and then develop into an organism made of trillions of cells? And basically there are 3 steps involved. We have cell division, which is basically mitosis, cells dividing. So you start out with 1 cell, the zygote when a sperm swam into the egg. Then it divides, now you have 2 cells, then 4 cells, etc. So here's cell division that makes identical daughter cells. All of these cells are going to have the exact same genome or the exact same sequence of DNA. And then you have cell differentiation. It wouldn't do us any good to be a ball of identical cells. Our cells are going to need to specialize. And so we call that cell differentiation, where they become different types of cells. So our stem cells are basically undifferentiated cells that can become anything depending on which genes are expressed and which proteins are going to be made in those cells. So really, when it comes down to it, cell differentiation is based on the proteins that are specific for those tissue types. And then we have, and which genes are expressed. And then we have morphogenesis, which is basically the body plan and taking shape. So with this in morphogenesis, it's where you can see, like, starting out with a single cell, and then you can see the eventual organism. Okay, so when we look at this, here would be like morphogenesis, like how do we get from a single cell to a developed human? And so when we look at the way it all starts is when we talk about like cell specialization, we're basically talking about different types of tissues and organs that are going to work together to have us be like specialized individuals. So with this, how do we get started, right? So inside of our egg, this is an egg before the sperm fertilizes it. These little circles represent like specific transcription factors or activators. And inside the egg, before the sperm even fertilizes it, it is not homogeneous, meaning they're not evenly spread out. This is significant because as the cell begins to divide, each of these daughter cells is going to be exposed to different transcription factors that will have a profound impact on the future development of this organism. Because these are transcription factors that are going to influence which genes are expressed and determine the ultimate fate of each of these cells. Okay, so inside of each unfertilized egg, there are pieces of mRNA and protein that act as these transcription factors. These come from the mom. The mom has special genes that when they are expressed, they code for these cytoplasmic determinants. So really, they're transcription factors or activators, but the fancy name is a cytoplasmic determinant because they're found in the cytoplasm and determine the fate of the cell. So with this, here's how it works. Because each of these cells' nuclei are exposed to different cytoplasmic determinants, the combination of these activators help determine which developmental pathway these cells will go on by regulating expression of the cell's genes during the course of cell differentiation. So here's a picture from the textbook just showing how they're not evenly distributed throughout the cytoplasm. Now, so a cell can start, the zygote can start with the transcription factors already in it, in the cytoplasm, but another way that can guide development is through the process called induction. Where induction, if you remember the 3 stages of cell signaling, is when a cell receives a message here, a signaling molecule, that can ultimately influence like gene expression. So you have a signal transduction pathway and an ultimate cell response. So in the case here of development, that cell response will be to activate transcription. So, and gene expression. So induction. Induction is when you have 2 cells near each other. So we'll call this cell number 1 and this will be cell number 2. So cell number 1 is going to induce cell number 2 to specialize. Now, how it does that is it sends out signals, local regulators close nearby, that will then lead to a signal transduction pathway where the final cell response is a turn on gene expression. So that right there represents like a transcription factor that will then allow RNA polymerase to transcribe and make the messenger RNA that then is going to make special proteins. Now, it's these proteins right here that lead to observable changes because each type of tissue will make special tissue-specific proteins. For example, like your muscle cells make, or your muscles are made of myosin and actin, which are proteins. Well, you're going to find that in your muscles. You won't find that in a neuron. So when we talk about tissue-specific proteins, we're talking about ones that will lead to observable changes in the cell. So to summarize, induction, these signaling molecules send a cell down a specific developmental path by causing changes in its gene expression that then lead us to see the type of cell that it will develop into. Okay, so this is a picture from the book. So when we look at how this happens, though, we have what's called sequential regulation of gene expression during cellular differentiation. It's a very long title, but basically what it means is that we're going to do gene expression. We're going to turn on genes in a sequence or in order to have our cells develop and differentiate. So as the tissues and organs of an embryo develop, their cells differentiate and they become noticeably different in structure and function. So here we start out with undifferentiated cells. We have stem cells that become lung cells or pancreatic cells or neurons or pigment cells or your muscle cells. All of these cells are noticeably different, and they're going to be different because of different genes that are expressed in each of them. So when we talk about a cell's fate being determined, it means that certain genes have been turned on and expressed, and it becomes irreversible. So in a stem cell, if there's certain control genes turned on for it to develop into a neuron, its fate has been determined, and it will become a neuron. There's no going back. Okay, so when we look at this, in order to have different types of cells develop, we need the expression of different kinds of genes. And therefore, we need tissue-specific proteins. Now, tissue-specific proteins and how this all works. We're going to use this as an example of a cell that will become or differentiate to become a muscle cell. So with this, we have what's called master regulatory genes or master control genes. So here we have a master control gene that maybe initially in the embryonic cell, it could either be from induction or the cytoplasmic determinants, those specific transcription factors that are going to activate this master control gene. So when that happens, the cell's fate has been determined. If this gene is activated, it produces a protein, and that protein actually happens to be another transcription factor. So earlier, when we saw the word sequential gene expression means that our genes are expressed in a sequence. So first, you have your master control gene activated, either through induction or specific transcription factors in the egg. Now, its protein product, once that master control gene is expressed, will lead to a series of events. Now, these proteins are all going to be specific, tissue-specific proteins that are going to lead to the differentiation of this cell becoming a muscle cell. So in this cell right here, its fate has been determined. It's going to develop into a muscle cell because this gene has been activated. And so now once that happens, more proteins will be produced, as you can see in this part of the picture, like myosin and actin, for example. And now as those proteins accumulate, that cell right here will develop into a muscle cell. And so that is really how master control genes work. And now tissue-specific proteins are only found in specific types of cells. So for example, if it was like a muscle cell, as you have these proteins accumulate, that cell will specialize and become a muscle cell. All right. So now, also during development, it's important to go through apoptosis at critical parts. So for example, we can see this in like a frog. As a tadpole develops, the tail doesn't just get cut off, but it actually is removed by apoptosis. Same thing happens with the webbing between our human fingers. As a developing our fetus, we have skin connecting our fingers, but as adults, we don't. Well, hopefully. And so cells can actually go through programmed cell death called apoptosis. So here are a couple steps involved. So the DNA and organelles. So for example, first off, this cell may be in the tail of the tadpole or in the webbing between our fingers as a developing fetus. But if it receives a signal to go through apoptosis or cell death, the first step, the DNA and organelles get chopped up. And then the second step is going to be the cell becomes lobed or goes through blebbing. And then the cell parts are packaged up into vesicles. And then you have scavenger cells will actually come and clean up and recycle these parts. So these phospholipids, carbohydrates, proteins that were in the cell will be cleaned up and reused. And it leaves no trace. And so here's just another picture showing apoptosis can happen while we're developing, but it also happens throughout our lives. So if a cell is damaged or stressed out, it can get a signal, like a ligand, to go through apoptosis. It starts to bleb, which is like folding in, and then it starts to break off into vesicles, and then your macrophages will come and recognize those and clean up the debris. So this is just a review of that first part. So now with this, our next thing to look at is setting up the body plan. So this little fruit fly is actually really well studied. This is Drosophila. So at this point, we're going to switch over and use the fly as our example to understand what's happening. So as we set up the body plan, we need to develop like spatial organization. Where is the head region? Where should we have wings? Where should we have legs? What about the abdomen, etc.? So when we talk about organisms, we talk about them in terms of like the anterior or posterior ends, and that's like your head to tail axis. Dorsal is like the back, and the ventral is the stomach. So we have different axes that we can reference. So as we talk, we're going to talk about like the anterior and posterior ends. So here we have cytoplasmic determinants in this egg for a fly, for a Drosophila. There's one particular cytoplasmic determinant called a bicoid protein. And you can see here that it's in high concentration on the anterior side. So here, this gradient, remember we saw earlier that they're not homogeneous. So this gradient of high to low leads to the expression of different genes that lead to segments forming. So with this, here we have our anterior end and our posterior end. So as this organism develops, wherever there's high bicoid protein, well, that will lead to the anterior development. Wherever it's missing bicoid protein, it'll be the posterior development. And so once we have these like different segments formed, what happens next is we need to know where to turn on the genes for like limb development and different anatomical structures. So here we have that bicoid protein. We'll turn on genes in the anterior region, and here we have something new. We call these homeotic genes. So these homeotic, these are transcription factors for homeotic genes in these different segments. Now, homeotic genes, or Hox genes, are what turn on like wing development, legs. Basically, they're turning on the genes for like the late stages of adult like form, I guess. So homeotic genes control the development of anatomical structures. So wherever you turn them on, so here, if that acts as a transcription factor, well, maybe this right here is a region. This transcription factor will turn on the genes that control antenna development.